start. So today we're going to be talking about chapter 8. We're going to be talking about diseases. Specifically, we're going to be talking about viruses, bacteria, and any other agent that actually causes diseases. It is important for all of us to understand that not all bacteria are harmful. In fact, there's actually a lot of bacteria that is actually useful, and we'll talk about that. So if you're ready, let's go ahead and get started. Microbes are microscopic organisms such as viruses and bacteria. They're found everywhere throughout the environment. They're found in objects. They're actually found in us. There's actually bacteria in the skin. There's actually bacteria in the gut. It is important for us to understand that not all bacteria are harmful. Some of them are, but the majority of them are actually beneficial to us. In fact, there's actually bacteria living in your stomach that allows you to break down the food that you consume. There's actually bacteria in your skin that actually prevent the actual harmful bacteria from entering into your body. And in fact, there's drugs that are actually produced by bacteria. Disease-causing agents, and this could be viruses or bacteria. Now, the good news is that our body has actually defense mechanisms in order to prevent for those viruses or bacteria to actually get into inside the body. And these barriers are, for example, the skin, which is the biggest barrier that you have, also the mucous membranes. And even if those pathogens actually get inside the body, our body actually has other defense mechanisms, such as, for example, white blood cells, T cells, that basically their purpose is to surround those cells that have been infected with the virus with an effort to destroy them. And the good news is also that our body is able to remember once you've been infected with a specific virus, so that way they can trigger an immune response. So pretty much every single time that you get sick is because it's a new different strain of a virus. Bacteria is a single-cell prokaryotic organism, and there's basically three common shapes. So coccus, which is sphere shape, bacillus, which is rod shape, and then spiridium, which is curved shape. Now, most of the bacteria, they actually have a cell wall, and the cell wall serves as a protection mechanism for the bacteria. Now, the cell wall is made out of a compound called peptidoglycan, and some antibodies, such as, for example, penicillin, so the way it works is that they prevent the formation of the cell wall. So they actually bind into the peptidoglycan, and if you will, it makes holes into the cell wall. And so what happens is that water can actually go inside of the bacteria, and the bacteria actually explodes. In some of the bacteria, they also have what is called a capsule, and the purpose of the capsule is actually to allow the bacteria to stick into surfaces in order to protect them as well. And it is important for us to determine what type of bacteria you have, so that way they can give you the right antibiotic. In order for us to do this, we actually add a specific dye, so we call this the Green stain. So if the cell wall has a thick layer of peptidoglycan, whenever you add that dye, then the bacteria is going to be purple, and so we call that bacteria gram-positive bacteria. If there's no peptidoglycan, then the cell is going to stain pink, and they're considered gram-negative. So these bacteria actually have an outer membrane, which is lipopolysaccharide, and it's actually released when the cells are killed by the immune system, and that stimulates actually inflammation and a lot of times actually fever as well. So again, it is extremely important for us to know if you have gram-positive bacteria or gram-negative bacteria in order to determine what antibiotics to actually give you. About bacteria, specifically, how does bacteria become resistant to antibiotics? So if you remember, when it comes to the bacteria, the DNA is basically flowing inside of the bacteria. So bacteria, they don't have a nucleus. And the other thing that bacteria has, it actually has also a small circular pieces of DNA that are called plasmids. And the plasmids are the ones that actually involve in giving the bacteria resistant to antibiotics. Now, one important thing about it is like if the doctor prescribes you antibiotics for, like, say, for 10 days, I know a lot of times what happens is that the patient actually feels better, like say at day number 5, and because they feel better, what they decide to do is like, you know what, I don't have to take the pills, for the rest of the time. And that's a really bad idea, because what happens here is that not all the bad bacteria are actually dying, and so you're basically adding a selective pressure for the bacteria to evolve. And essentially what happens is that the bacteria is going to become resistant to the antibiotics. So that's why it's extremely essential and crucial for us, if the doctor prescribes you antibiotics for 10 days, you should take them for 10 days. right? It's called binary fission. And essentially what happens here is that a single bacteria is split into two. And this is why, within days, within hours actually, you actually have lots of bacteria. Again, because the way it happens is that a single bacteria actually can divide into two by the process of binary fission. Are super small, they're very small, and they are considered intercellular parasites. They're considered not to be alive, and the reason for that is because they're unable to maintain homeostasis, and a virus doesn't have the machinery to reproduce. And this is why a virus actually infests a host cell with the goal of stealing its machinery to make more viruses. Now, a virus consists of two parts, another protein called caspid, and then inside the caspid you actually have either DNA or RNA. And DNA and RNA actually carries the genetic information in order to make more viruses. Again, a virus is considered not to be alive because it cannot maintain homeostasis, and it basically doesn't have the machinery to reproduce, so that's why it infects a host cell in order to make more viruses. So here this shows an image comparing the three types of cells. You actually have a eukaryotic cell, you have a prokaryotic cell, and then a virus. So what you can see here is that the virus is super small in comparison to a eukaryotic cell. Virus attaches to the whole cell. What's going to happen is that the virus is going to inject either DNA or RNA, and that DNA or RNA is going to have the instructions in order to produce more viruses. Specifically, what happens inside the host cell is that the virus uses the ribosomes. The ribosomes for the machinery that the virus uses in order to produce more viruses. Image of a virus. So what we can see here is that you can see like the caspid, which is made out of proteins, and inside the caspid you actually have either the DNA or RNA. And on the surface of the virus you actually have other proteins. And the important thing is that those proteins actually resemble the proteins that are found on the host cell, so that it's easier for the virus to attach and inject its either DNA or RNA.